Tonight is Clay Bryce. Clay has been a member of the WA Museum's Department of Aquatic Zoology, worked at the museum for over 35 years, and there's an awful lot of people like Clay who have found a niche in the museum, and there's no reason to actually go anywhere. The accumulated experience in the museum scientific department is immense. And Clay's participated in many marine biological surveys in the Kimberley region. He's currently heading up a major project examining the biogeography of the marine fauna in the northwest of WA, with a particular emphasis on the Kimberley region. Clay also established and ran the WA Museum documentary unit for several years in the early years of the century, where he produced and directed and co-wrote 13 domestic and international TV programs. So that's enough from me. Without further ado, I'll introduce you to Clay Bryce.
Well, thank you very much, and thank you for coming out on this, I guess for Albany, a balmy evening, but for me it's a tad cool. I'm going to battle with technology tonight. We're going to start with me talking, and then I'm going to switch to a documentary. It's an 80-minute documentary that looks at Scott Reef, which is one of the outer edge atolls out from the Kimberley coast, and that has a particular story and a particular relevance to the whole survey work that we're doing in the Kimberleys. So, collecting marine life in the Kimberleys. If I get this right, it'll be great. Let's see how we... Oh, okay. So, I thought I'd start off with where is the Kimberley, because on a lot of these lectures that we do give, there's quite a few people turn up that have no idea where it really rests. Where is the Kimberley? It sits at 129... Oh, actually, excuse me for a minute. I've got a fancy thing here. There we go. Look at that. We have actually the Northern Territory border, which actually runs along the old Pope line of 129 degrees. This line here, while at 19 degrees 30 minutes, is actually sawtooth and jagged because of state electoral boundaries. So it does actually go on a straight line. It's sort of very, very jagged. And it was named after the Earl of Kimberley when Alexander Forrest visited the area in 1879. And it's a big area. It is huge. It's 424,500 square kilometres, twice the size of Victoria. It's got a monsoonal climate. It's one of 15 national hotspots. There's approximately 2,500 islands to 3,000 islands. And we say that because it just depends on the tide. The population is very sparse, 12.5 people per kilometre. And if you look there, you see the Australian average is 35 people per kilometre. And to put it into some sort of perspective for you, in UK the population is 3,075 per kilometre. So there's not a lot of people in Kimberley at 12.5 people per kilometre. I can never work out how they do the 0.5, but there you go. So, the Kimberley, the marine life in the Kimberley. The marine life is there by virtue of some major drivers, environmental drivers that drive the whole system. If you look at the currents, you have what the newly described Holloway Current, which is seasonal. It runs around about the end of March, at the end of the monsoon season, and it's a surface current. Not a lot is known about it, but you will note that it goes past Scott and Stringham Reef and Mermaid Reef. I've marked those black spots on myself. You can see they're very professionally done. There's also this huge Indonesian through-flow, which is a body of water that just flows through the islands, comes around this main gyre here, and pulls itself back into the coast and then joins up with the Lewin Current. This ensures that most of the west coast, including the Kimberley, has mostly... well, in the majority, it's Pacific fauna. It's not Indian Ocean fauna. You do get some Indian Ocean species, but few, given the percentages. Down here, of course, you get a lot of endemic species. Few endemic species, but mostly Pacific. The coast is very wide. You see you have a very wide continental shelf coming out here, out to the edge of the shelf here. So that ensures that the spread of animals coming down here is sort of well spread, especially when you have cyclones that come through, which is the other main driver. On the land you also have fire. We're not sure of the effect of fire, but we know that there's an awful lot of fire going on up there with prescribed burns and natural burns, and all the ash and nutrient-rich debris that goes into the waterways and flows out to the rivers, we sure has an effect, but we don't know what, and it's a concern of great worry. So, in close to shore, we have heat, desiccation, scouring wave action and sedimentation all have effect on the reef flats and the animals that live there. as well as a freshwater inflow with regards to turbidity and nutrients, and that's mostly from the cyclones. And we'll get onto the cyclones in just a few minutes. Right now, we'll get onto the cyclones. From 1906 to 2006, that's the cyclones we've had. It's kind of amazing, really. But we get more cyclones than anywhere else in the world here. And if we go from 2000 to 2006, you can see how the numbers are building. It's a little bit more clearer there compared to over here. In 2003, 2004 cyclone season, Cyclone Faye came roaring across the top of WA, went right across the top of Scott Reef with 300 km/h winds. Did a huge amount of damage. We'll talk about Scott Reef a little bit, but it's just to sort of show that cyclones have an immense effect on this coast, especially given the field-like coastline, the big tides, and this huge surge of water that comes in here and pushes down these narrow channels. It has a huge effect. Scott Reef had these huge boulders were thrown up on the reef. The waves were so big. These were live corals that were further down the reef slope. Coral bleaching is another thing that's affecting the Kimberleys. It's affecting the whole of the tropical world's oceans. And at 1998 and 2003, it hit the Kimberley pretty hard, especially out as far as Scott Reef. And we'll get onto that again in a little while. So we have warming of the oceans through anthropomorphic disturbances. We've got the cyclones, which come through and do huge damage, and again their intensity and their severity is increasing. And then we have, well, this is an example of some of the damage caused by the coral bleaching, where the corals were all died, then Cyclone Faye came through and smashed it all to powder. But we do have examples of regrowth coming back, which are these bits here. The other main driver is unsustainable fishing pressure, mostly from Indonesian fishermen due to their being allowed to fish up off Scott Reef and String of Batam. This is all clam meat, giant clam meat here. So, what has the museum been doing up there? As far as the offshore area, now that's the area in red versus this inshore area. So as far as offshore, we did the Rolly Shoals in 1982, which is down here. We did Scott and String of Batam in 1984, Ashmore and Cartier in 1986. Then I did 3 informal surveys in 92, 94 and 96 covering all of these. Then in 2006, I did a resource survey up here, marine resource survey up here. And then we did Mermaid, Scott and String of Batam in September of 2006. These guys here in red were all done in 2006. Now, before 2006, we're calling historic data. After 2006, we're calling it more contemporary data. It's because our survey methods changed. We suddenly became, I guess, more scientific. Instead of doing what our Act told us to do, which was to go up and survey the marine biodiversity, we actually went up and started doing counting and a little bit more hands-on science as to why the animals were really there. So that marcation there is quite important, and that'll come out a little bit further in the talk. Inshore, we did Admiralty Gulf in 1976, Broome to Cassini. In 1988, Broome being down here, Cassini Island just up here. Wynnum to Broome in 91. Eastern, Southern and Central Kimberley in those years, and Eastern Kimberley is here, Central Kimberley and Southern Kimberley. We did in those 3 years. Beagle Bay in 97, which was a small survey for a pearl company. And Adele and Montgomery Reef in 2009, which was just last year, and this was Adele and Montgomery Reef. Oh, sorry. And in October this year, we will be going back to Long Reef and Cassini Island to resurvey those. And that's part of our more contemporary surveys, where we're doing a reef and an island, and a reef and island. And then we're also proposing to do more field work in the next 4 years through this area here and out as far as Browse. But that's for the future. So what have we been looking at? We've been targeting the crustaceans, we've been targeting the conoderms, the molluscs, the fish, the corals, algae and seagrass, worms and mangroves. That's a lot of people on a boat if you look at 1 to 2 people looking at each group. The mangroves are a particular interest in the inshore Kimberley, and it's a lot of fun playing in the mangroves. You get to sort of climb through them, being watched by crocodiles. You get to climb up mud slopes, watched by crocodiles. We also have diving in the Kimberleys. We've done a fair bit of diving in the Kimberleys. It's seriously spooky, as you can see. You don't know what else is down there with you. And we have problems getting in through narrow channels where it's very, very shallow. If you get stuck, you're there for a long time. We have engine breakdowns, and we've had several of those. We've had major breakdowns. The guys have been covered up just to stop fan mail. So there's dealing in remote areas like the Kimberleys is not easy. It's fraught with all sorts of problems, and there's all sorts of unexpected problems which you just have to deal with. That's just the nature of the beast. Admittedly, in more recent times, especially since 2006, life is easy. We can get helicopters to come out, we can get other boats to come out. But in 76 to 94, it was a different story. It was pretty remote. So what were the outcomes from all this work we did up until 2006? We had 10 reports, we did 2 major publications in book form. We looked at 9 different taxa, that's the different phyla of animals that we targeted. We visited 158 localities. We're still counting under this new program I have running at the moment, which I'll talk about in a while, but we're still counting the number of species or specimens that we collected, voucher specimens in the state collection. At the moment we're well above 20,000. And the number of species recorded itself are in the thousands, and as I said, we're still counting. It's a huge amount of data that's come in from those early trips, and we're still coalescing all that data and seeing how we can analyse it. So in 2006, things changed. Everything sort of, just all changed. It all got really weird. We formed a WA Museum survey team, which was an ad hoc team in name, but what it meant was we would bring in people from all over Australia and the world if we didn't have the expertise ourselves. So we would get a really big boat with a lot of people on it who had the expertise to go up and look at the animals that need to be looked at. And this sort of produced a lot of data that we're working up now, and some of it's already been published. We also formed a partnership with Woodside Energy, who have been very, very good to us in providing funding for research and development with no strings attached, which is very, very nice of them. And that's allowed a lot of this work to be undertaken, not only what we're doing now, but also in analysing the data of the past. So in February 2006, the beginning of the change, I went up and did a marine resources survey of Scott Reef. Now the marine resource survey is for holothurians, trochus and the giant clams. These are the species targeted by fishermen. So the idea was to go up and see what has changed at Scott Reef from previous surveys done by CSIRO and other agencies. It was looking at the invertebrates only, because that's what they are, only invertebrates, and only shallow water habitats. These are the stations we looked at, and these are long transects. Some of these transects are kilometres long. We did 19 transects covering 16.8 kilometres and a total survey area of 6.8 hectares. In 8 days, that's a serious lot of swimming, I can tell you. Is that right? That's what we did. That's fine. So what I'm going to do now is I'm going to attempt a technological wonder and try to switch to the video, because that survey that we did was out at Scott Reef. So now I'll show you a video about Scott Reef, and you can see some of the problems out there, and you'll have a bit of a rest from me. So I'm going to chicken out and go this way because I think it's easier. And then I'll have to go, bear with me for a minute. Is there a cursor here? Where? Is it? Is there a cursor? Yeah, but I can't. Oh, there, but it's not working. It's not, oh, there it is. Sorry. Yeah. And then we want. Look at that. Amazing. And then I'll have to go full screen. No, I don't have to press that, don't I? This is very tricky. Oh, get out of your way. Is that in your way?
Scott Reef Atoll is remote. It lies some 200 kilometres off Western Australia's Kimberley coast. Like a desert oasis, it is a hub of life in a wide tropical ocean, where marine life and the forces of nature have been in balance since its very beginning. But over the last few centuries, the pressures of man, raging cyclones and coral bleaching have combined to cause an upset that has shaken this marine paradise to its very core. Scott Reef sits in the centre of a chain of deep water atolls and reefs, stretching some 1,000 kilometres along the edge of the Australian continental shelf. These oceanic atolls, with their central lagoons, have massive protective walls formed when skeletons of corals, algae and other calcareous creatures combine with accumulating sand and rubble. They are complex and dynamic environments. Scientists have only recently begun to study and fully understand them.
There's the very familiar to us all shallow reef that we typically dive on or walk on at low tide, but either side of that emergent rim there are deeper areas of habitat going down 30, 40, 50 metres still with algae and coral that you associate with reef and with fish. It's quite abundant down there and somewhat sheltered from the disturbances of bleaching and storms. Then the reef drops down over the edge into hundreds of metres of depth, where light is far less important and a completely different type of functional ecosystem operates.
The reef platforms of Scott are mostly submerged, and the entire reef is made up of 2 atolls covering an area some 580 square kilometres. North Scott is an annular or round atoll with a shallow lagoon in the centre, while South Scott, about 3 times larger, is horseshoe-shaped and open to the north. Its lagoon is very deep, making it unique among Western Australian atolls. South Scott also has a sandy islet separated from the western horn of the horseshoe by a deep channel. Called Sandy Island, this piece of land has no vegetation, but is home to nesting turtles and breeding noddies. Migrating seabirds also use the island as a resting stop. Scott Reef's massive walls rise steeply from the deep ocean floor to almost the surface. Here they form a barrier between the crashing ocean swells and the quiet waters of the lagoons. As huge waves pound the outer side of these walls, their energy is released and reduced to almost nothing as the water flows across the exposed reef and into the lagoon. As the waves pass over the reef, they cross the main habitat zones. The reef front slope, the platform, the back reef, and then into the lagoons themselves. The quiet waters of the lagoons, the turbulent outer reef and the depth of water provide the basis for a range of habitats which merge to form a complex ecosystem.
It's a bit like looking at a painting in a museum. The harder you look, the more you see in it. When you start looking at the data, and more importantly, using your eyes and really absorbing the environment you're in, you start to see a lot more complexity, a lot more detail, a lot more pattern.
Large areas of the reef platform or reef flat are exposed at low tide. Here the sun and wind provide a desiccating force for any life form not adapted to this harsh place. Rock pools become sanctuaries for fish, corals and other invertebrates.
Within there, you'll find specialist groups of corals, algae and so forth. For example, with the algae, you might have fleshy algae doing well towards the more sheltered areas of the reef flat. And often out where the waves are pounding and the reef flat begins, very strong encrusting and often beautifully coloured pink calcareous algae tend to dominate.
Either side of this massive barrier, the habitats are noticeably different from each other. This is where the reef begins to break away, forming nooks and crannies for marine life to hide. On the outer edge, we find deep surge channels and tight caves that have been sculptured into the reef by the constant wave action. In this turbulence live a multitude of fishes, corals, and other small animals. Deeper down, the water becomes cool and dark. The reef slope lessens and gives way to scree and rubble covered in fine silt. Life here is less frequent and only the very hardy survive. In contrast, the inner edge of the reef platform, the back reef slope, is a maze of flat-topped coral bommies and interlocking pools. Here the water is shallow and relatively still. This is an ideal place for marine life dependent on sunlight. Encrusting and free-swimming animals find shelter amongst the large formations and broken coral rubble. Another distinct habitat occurs at North Scott, where two drainage channels cut through the reef, linking the lagoon with the open ocean. As the tides ebb and flow, these channels turn into a highway for prowling fish. The flowing current delivers food to filter feeding sea stars while refreshing and flushing the lagoon waters. While North Scott Reef has its drainage channels, South Scott has a very deep lagoon to mark its uniqueness. Until recently, this lagoon had been little explored, but by sending down a remotely operated vehicle with an onboard camera, scientists are now able to study this deep water environment. What they expected to find was coral debris and silt washed down from the shallows. However, the camera has revealed a mix of delicate foliaceous corals, rubble fields and meadows of calcareous green algae called Halimeda. These deeper reefs appear to be little affected by recent external forces. The same, however, cannot be said for the shallower reefs. In 1998, a devastating coral bleaching event hit many of the world's tropical oceans, and Scott Reef was no exception.
Coral bleaching is a phenomenon where the microscopic algae in the coral are disassociate and either abandon the coral or lose their pigments, and the coral is still alive initially in this stress response. But what happens is that without its pigments and its algal pigments, you can look straight through the tissue, which is like looking through a jellyfish straight over the limestone skeleton. So the coral looks white because the skeleton is white limestone.
This bleaching occurred when unusually warm seawater washed across the reefs and stayed for a prolonged period. The rise in temperature caused many of the corals to go into stress.
Now, if that stress stays there long enough, the corals simply run out of food and die. If, however, it's just a short-term stress, there's a good chance that the coral will get its algae back working properly in a symbiotic relationship and recover. However, bleaching does take a toll on the resources, the lipids and so forth involved in the coral growth, and whether it kills them outright or it's only transitory recovery, there is an impact ecologically.
After the coral bleaching event in 1998, over 80% of the shallow water corals on Scott Reef died. But bleaching is not the only force of nature that these oceanic atolls must deal with. Between 1988 and 2004, 20 Category 4 and 5 cyclones pounded this region. The northwest of Western Australia has the highest incidence of cyclones in the southern hemisphere. In 2003, Cyclone Faye unleashed her fury. She roared into the Timor Sea, reaching maximum intensity directly over Scott Reef. Screaming 300 km/h winds generated huge seas which smashed into the reef. For coral colonies and other life forms regenerating after the bleaching event, Cyclone Faye provided yet another challenge to their survival. The team from the Australian Institute of Marine Sciences visited Scott Reef shortly after the cyclone and saw firsthand the power of nature.
The first thing they saw were massive boulders, car-sized lumps on top of the reef that weren't there the last time they visited. Now, on closer inspection, climbing right up to these things, they realised that these big lumps weren't just rock, they were actually formerly living coral that the guys had been diving on 6 to 10 metres down the reef base. So the storm rolled them all the way up and plonked them on the reef.
All around them, the habitats of Scott Reef had been devastated.
The fragile reef system that had only begun to recover from the 98 bleaching was now just a trail of broken branches and grey algal-covered rubble.
But the effects of cyclones and coral bleaching are something the reef's natural resilience can withstand and recover from. When fishing of its marine life runs unchecked, however, especially across a recovering reef, the balance of nature has little chance of returning to normal. A recent survey by the Western Australian Museum paints a dim picture.
Since the early 1880s, Indonesian fishermen have been coming down to the reefs collecting marine resource species, which are the holothurians, the biche de mer species, the giant clams or the tridacnid clams, and a single large gastropod mollusk called Trochus niloticus, which is just occurs on the very edge of the reef. And over a long period of time, those populations have been declining. February of 2006, the museum did a survey of the shallow reef to see just how many of these animals were still there, and we found to our great disappointment that the population had actually crashed. So many individuals have been taken off the reef that there's just not enough there to reproduce in any sort of meaningful way for the future. So it could take decades, 50 to 70 years for these reefs to come back unless some sort of sustainable management is put into place.
Today, Scott Reef bears the scars from the combined impacts of bleaching, cyclonic activity and fishing. Long stretches of reef and lagoon pass by festooned with regenerating corals, but there is no sign of giant clam or vacuuming sea cucumber. There is still much to learn and understand about Scott Reef and its many life forms. How it appears today may not be how it will appear in the future, as the reef is a constantly changing ecosystem, adapting to the demands of the prevailing environment and the impacts of man. With the technology to study life forms at great depths, scientists will continue to build a picture of this complex oceanic reef system.
The outside edge of the reef drops off to hundreds of metres, and the fauna down there is completely different. These go right down to abyssal depths, and it will be exciting to sort of make an inventory of the marine life there and see how that interacts with the shallow water species.
The research for the future will be to understand the natural dynamics of the reef and get a far, far better understanding of its overall resilience and the things which tie it all together, many of which probably have been hidden from scientists in the past because we've tended to be a little bit seduced by scuba and focused on those beautiful shallow water areas. But in the future, I think we'll be taking a more holistic approach and looking at the reef as a functional ecosystem in its entirety.
So, that was Scott Reef. If you just bear with me and I'll see if I can... I forget how to do this. Oh no I don't, I go there don't I? Yes. And over there. Just a point of interest. Actually that's wrong, what happened? Just as a point of interest, we shot that whole documentary by snorkel because we weren't allowed to use scuba. Which is a lot of fun on those scooters, I tell you, it's really good. So what did we find after we went up and did this survey, this marine resources survey at Scott Reef, when we went up to look at the marine resource species? We actually found that in 1998 CSIRO found 15.8 commercial holothurians per hectare, 10.5 tridacna per hectare and 0.03 Tectus niloticus. When we went up in 2006, that's the reduction. You end up with such a dilution of animals that any sort of meaningful reproduction is almost impossible through oligospermia. It's just not going to work. Just as a note, these are used as food, and mostly for mother of pearl inlay. So it's a little bit sad, but there you go. So in 2006, later in 2006 in September, we formed the Marine Survey team and we had to go up and do our first major marine biodiversity survey. And that's not looking at the marine resource species, that's looking at all animals, of all the major phyla, which is those ones we targeted earlier, the crustaceans, the mollusks, etc. So we formed a team and we went out with the aim to record the species that occur at these three northwest atolls, to determine its distribution and abundance, and to compare the results to the other studies. And the other studies are the historic data. So if you remember back in 84 and 86, we visited Scott Reef and Rolly Shoals in 82 and 84 and 86. So it's some 20 years later, we've had a chance now to go back and see how things have changed. We did 17 stations at Mermaid Reef, 23 stations at Scott Reef, and 5 stations at Seringapatam. The results, I'll go through briefly the results because they're fairly boring, I suppose. But what's important, I think, to remember is that there was some 1,500 species and over 128 records, new records. And what I mean by a new record, for those who aren't familiar with it, is that when they say one new Australian record for molluscs, that means that's the first time that species has been found in Australia. First time, there's 3 species for the first time recorded for WA, and for the reef itself, there was 2 species. And again, most of the molluscs are Indo-West Pacific species. For the sponges, we have these sorts of figures: 132 species, 79 species unique to the reefs. In other words, they're found nowhere else in the world but those reefs, which is phenomenal. For the corals, there was 269 species, mostly widespread Indo-West Pacific species, and again, an incredible 22 species never recorded before, another 18 species for Seringapatam, and 1 new state record. Now, these are things that haven't been recorded before, since before our trip. So there's a lot up there that still we don't know. Every time we go up, we find more and more. The echinoderms, the starfish, again, same sort of a story. The crustaceans was 157 species, which is double the previous known records for the area, and that's massive. That's a huge increase of species that we didn't know were there before. Fishes, again, 461 species, 1 new record. Given fishes are usually well known, just finding a new record is quite significant. And the marine flora, 121 species. This is the first detailed account for marine flora. And it was 24 new records for WA and 4 new Australian records. And the whole effort was published in this volume in 2009. It was put out, and it's available on the web. If you just dial in WA Museum, North Australia, or Kimberley, you'll get to the website and you can go in and just print that off if you want it. So, what are we doing now in the Kimberleys? In 2008 to 2011, we started another program where we were going up in conjunction with the Museum and Art Gallery of Northern Territory, the Australian Museum, the Queensland Museum, and the Herbarium. The idea to look at broad scale, look at all the historic data we had previous to 2006, to get all that data together, all those databases together, which has turned out to be a job bigger than we ever thought possible. It's actually quite scary, the amount of databases we're having to deal with, and we're not sure we can cope, but we're doing our best. To look at all that data in a broad scale way and say, well, what story does it tell us? We also did 2 trips, one to Adela and Montgomery in 2009, and one this October in 2010 to Cassini and Long. That's fine scale. That's where we're using the new survey techniques. So we have this old data giving us a broad scale, broad brushstroke sort of look, and then the fine scale stuff. Oh, wrong way. So with the historic data, to give you some idea, you have the wet collection, which is here, the dry collection, and these 2 guys here, these 2 intrepid workers are going through box by box, jar by jar, vial by vial, recording all the data on databases that come from the Kimberley. They've been at this now for 2 years and are seriously going stir crazy. But they are stoic. They are. And there's a high degree of accuracy needed, so you can imagine what they're going through. So far, they've done 19,000 lots, which is an incredible number, you know. So formatting all this data from the Australian Museum, we got polychaete data, we had soft coral data, crustacean data, marine flora data from the Herbarium, and from the WA Museum, because this is our state, we had all of these groups here. And this represents hundreds to thousands of species that we're trying to put together. It's a huge job. But it is showing us some interesting pictures when we do some multivariate analysis on the data, which is kind of nice. So right now, as I said, we were looking at these reefs here. So we picked a reef, and again a reef and an island to look at some sort of latitudinal separation. The data we're getting from here will then be compared with the data we're getting out there to give us longitudinal separation as well as this latitudinal separation. So we're quite serious about how we're going to analyse this data. Again, we're targeting the same groups that we did in the historic data to give us some sort of context. But how are we going about this? Adele Island. Adele Island has never been surveyed before, ever. It's never had any surveys of any sort done on it. 1948 was the last survey, and that was a desktop analysis of aerial photographs from World War II. And that was to look at just the geomorphic units. Adele Island is an amazing place. It's sandy with lots of spinifex in the middle of the island. huge bird colony, including these frigate birds, the noddies, boobies, sorry, these are noddies. And we did a lot of diving and working on the reef flats to get the data that we needed. Montgomery Reef was a whole new ball game. This is 300 square kilometres of reef flat that comes out of the water. To deal with it in a survey, given the limited time and resources, was kind of scary. So to do this, what we did, we divided it into geomorphic units. You have what we think is this central terrace. Then we have these lower terraces. Then we have this front reef ramp that runs all the way around the edge. This area here, we decided to leave for another day because it was just too big. The whole area is just too big. So if you get a closer look, this area here, you can see the water charging out of here as the reef comes out of the water. This is your fore reef. This is the lower terrace. And this is what we thought was an upper terrace. Again, this is the fore reef ramp as the water comes, as it drains out. And up on the upper terrace, you see this line here. This line here is actually a dam wall, if you like. And what we thought was the upper terrace has turned out to be a very shallow lagoon. And we didn't even know that's what it was when we went up there, but that's what it looks like it is. The lower terraces just drain. This upper lagoon never drains. Before it drains, the tide's on its way in again. It just doesn't have there's so much water up there to drain out. It's mostly the dam area is mostly made up of rhodoliths, which are calcareous algal balls, sand and coral rubble. And the whole area is moved. As you get a cyclone, the ridges of sand and rubble will move and then come back. That's just on a normal tidal cycle, it's slowly moving. So expose old coral behind and cover new coral. So it's a really dynamic place. It's constantly changing. We did 27 stations over 2 reef systems for both Adele and Montgomery. The 2 reef systems being Adele and Montgomery. We looked at 8 taxa, including video transect analysis. We did 324 researcher hours actually on the reef, not counting what we do afterwards. And we found a new species of fish. Some research we're doing now looking at the data means that we've probably got 4 new species of crustacean, but that's yet to be confirmed. A new species of coral, 2 new species of brittle stars, and we don't know how many records yet. We're still working our way through the data, which we hope to get done before we go on the next trip, because otherwise it's going to be really busy towards the end of next year. And so that's what we're doing. In the future, we're applying for money to go up and fill in those areas between the very north and the south, as well as out as far as Browse Island. And that will then help fill in from the outer edge to the coast, and also the latitudinal shift between Montgomery and Long Island. And thank you for your time. If there's any questions, sorry.
You organise questions?
Yeah, any questions?
Yeah, you mentioned that they didn't allow you to scuba dive when you filmed that. That you had to use snorkel?
Yeah, it was commissioned by Woodside Energy, and because the area is designated as a petroleum lease, to dive, the only way we could dive was to do hard hat on surface supply breathing apparatus, and it's impossible to film animals with all that gear on. It's too hard. Scuba we could do it, but not any other way. So the only way we could do it was to do it on snorkel. And then we discovered these fantastic things called underwater scooters, hands-free underwater scooters. So you could hold the cameras in your hand and just go where you want to go. Not only was it great fun, but it was we got very fit. You know, you can imagine diving 8 hours, snorkeling 8 hours a day for 2 weeks. But yeah, it was an excellent way to do it. But that was the reason why, because it was a petroleum lease.
I'm just curious, diving up there because of the crocodiles, especially in those channels through that Montgomery Reef system. How do you go with crocs?
Oh, I can be very flippant and say you smack them on the nose, which is what I tell a lot of people, but no. We try to avoid diving actually in the reef, in the river systems. We'll go further offshore and we check the area very carefully. Most of the diving was done in the years in the early 90s, but the crocodile population has increased exponentially since then. So this round of work we're doing up there now, we will only dive on the very outer reefs. where there's no chance of having a real problem. In reality, the biggest problem is sharks, to be truthful, and the currents themselves. They are the biggest problem. But all the divers are commercially rated, they're all very experienced, and we're very, very strict on our diving protocols. So no, we haven't lost anybody yet. It's very good to hear. Yeah, just imagine the paperwork. Yes, sorry? Sorry? We can, but you can drop all the cameras you like under the water, but you can't really identify the animals that way to species level. It's very difficult. You can get an idea of what's down there, but to identify species level is fraught with all sorts of problems. And then you have the problem of voucher specimens. If we collect something, we don't know what it is, we've got it to compare with other animals. If we collect a new record or anything of significance, we've got the animal in the collection, the state's collection, to compare and say, well, okay, when we produce the paper, we can back up what we've said is what, because we have the specimen. You get a lot of papers you see published nowadays where they'll say we collected all these animals, well, we recorded all these animals, but there's no veracity in the data because you can't prove that what you saw is what you determined was that species. And so that's something the museum is very strong about. When we say something is something, that's what it is, because we can back it up with a specimen to say that's really what it is. And if you want to argue the toss, well, here's a specimen, we can prove it. But so drop cameras have a role, but they're very difficult for us to use. Yeah.
Talking about the differences between drop cameras and videos, how does it sort of match up in terms of information you can get from...
You mean as we're doing this? Well, the videos we shoot are mostly for... Are you talking about the habitat video footage we shoot, or this sort of Scott Reef stuff we're shooting?
I was talking about the transects you're shooting on video.
Yeah, we shoot video analysis, and that's a matter of walking along at a set pace along the transect, and then that's analysed for gross habitat changes or the main phyla changes where you may go coral, soft coral, algae, sand, rubble. We do that. So drop cameras, it's easier to dive. It really is. You get a much better flow, especially with the currents. Trying to keep a camera on course if you've got a cross current is just really hard. Sorry, it's really hard for me to see you people.
You yourself dive, do you?
Yes, I do. I'm the chief dive officer as well. Yeah. Excellent. Thank you very much.
Interesting and fascinating talk and just a look and some areas that we would otherwise actually never see. If you do want to see this again, and obviously we've been filming this evening, but there are video vodcasts of all of the Wild West lectures on the museum website, and that's the address that Clay's put up for us there. So please feel free to go and have a look at this one and other ones that have been filmed over that series. And if you do know of anybody that couldn't get here tonight but would be interested, there is a repeat of this lecture tomorrow afternoon at 2 o'clock in this place. So encourage people, if they wish, to come along tomorrow afternoon, and Clay will be doing it all again for that. So thank you very much for your time. You're welcome to stick around for a little while if you wish to have a brief chat to Clay or to each other, and have another glass of wine. And thank you very much for coming, and we'll see you again. Thanks.